people sweat, skin conductance changes, and the signal degrades. So we need in this field to develop implants that go inside the body to get these signals more robustly. So within the center, we are now researching how we can grow nerves through what's called a microchannel array. So this is a large tube with many small tubes inside the large tube. And the small tube's on the order of 100 microns in diameter. And each small tube has electrodes to either sense descending commands coming down from the spinal cord to get the person's desire and how they wish to move, motor intent. And other channels will be sensory. So in those adjacent sensory channels, we can stimulate, reflect sensors on any external machine information onto the nerve. So effectively closing the loop between the human and the machine. So this idea exploits a robust biological phenomenon. If you take a muscle here shown as M, and a nerve that's been transected shown here as N, and you put them in close proximity, the nerve will grow, regenerate, and attach to the muscle. It's a very, very robust phenomenon. You can see the sprouting from N towards the muscle M. So on the other side of this array, we're putting the person's own muscle cells and skin cells to promote growth of the transected nerve through the array channels. In this experiment, we demonstrated that basic capability. So in an animal model, we have a transected nerve, and we put skin and muscle cells from the animal. The nerve grows, innervates, attaches to those end organs. And then the body of the animal quickly vascularizes the tissue, and one ends up with a very stable implant. So using microfab, we're building these compliant, mechanically impedance-matched arrays. Each channel has platinum electrodes with leads coming off. They'll go through to a bundle of wires, and that bundle will go right through the skin. So substantial nerve regeneration has been demonstrated through the array. This is immunofluorescence reveals axon regeneration after 3 months through microchannels with platinum iridium electrodes. Here, motor axons are shown in red, sensory axons in green, and Schwann cells in blue. So how do we get the information from inside the body to outside the body, and from the outside of the body in? One would expect the approach would be wireless communications, but I actually don't believe in wireless communications for this example. I think the future is wired. So here's one example of how that might be done. So to transmit nerve signals to and from an external bionic limb, one can do it through a conduit. So what you see on the left side of the screen is a titanium shaft that goes through the skin membrane right into the residual skeleton. This is of a kitty cat that lost a hind limb. Then you can bolt the prosthesis right to that titanium shaft and apply loads directly to the skeleton. One can make that shaft hollow. Guess why? To run wires right in and out of the nervous system, right into nerves and muscles and out to the bionic limb at extraordinary bit rate. So how can that be done without problems with infection? So what researchers are pursuing, this is a collaboration with Mark Pitkin at Tufts, they're searching for optimal geometries of synthetic materials where the animal or human's skin cells adhere right to the synthetic surface, creating a seal so one doesn't have an ingrowth of bacteria. One can also play this game not with an osseo implant, but just running wires through the skin and having a port right on the body. So I think in the future we're just going to have these neural buttons, and we hook up any machine to them, and it's a direct communication to the nervous system. Why is this